Everybody say hi. Let's see, I know it may be actually it's today. Angel may be the only guy who's not in class. But in case you were to be sick, you can watch the videos. Have you watched the videos at all?
No.
No?
I look at the PowerPoints, but I haven't watched all the videos.
The videos are exciting.
Yeah, I watched a little bit of it, but like my other professor, like literally hammering, like cramming stuff down our throats, so.
I can do that. No, I'm not much into cramming. Well, and watching lectures versus sitting here real time, I don't know how well it works. At the same time, I kind of get blown with PowerPoint, but this is what we're doing. Anyway, what we've been doing, of course, is going through the cell. And a good place to sort of organize your thoughts is looking at the definitions of all the bits and pieces in the cell, what they're doing, and the difference between prokaryotes and eukaryotes. So we've got then, we're just to back up a couple slides, our genetic elements, and I swear there was a way we used to be able to kill that light. But all right, so the nucleoid, this is the chromosome where the DNA is, and it's not in a nuclear envelope. And then we also have something that is really not entirely unique to bacteria, but most of their cells don't have it, and that is these small circular DNAs are called plasmids. Yeast can actually hold plasmids a little bit, but animals, we can't maintain a plasmid. When our cells divide, we don't have a way of divvying up or replicating a small circular non-chromosomal piece of DNA. There are cell culture techniques where you take animal cells and tissue culture and you can blast plasmids into them, but the DNA isn't replicated, and then as the cells divide, the DNA is being diluted out and sort of split up, and fairly quickly it just sort of degrades and disappears. But in bacteria, they can hold a plasmid quite well. Generally, in order to make sure that they maintain the plasmid, at least in molecular genetics, we've got some kind of antibiotic resistance gene on there as well as whatever we're monkeying about with. And you grow and maintain the bacteria in the presence of an antibiotic. But in the wild, there are plasmids that carry antibiotic resistance, and they can be transferred back and forth. And it's a pretty easy way to transfer DNA from one cell to another. All right. So then we finished up with some more general ideas about the cell and bacterial cells, and that is the idea of chemotaxis. And this is not entirely new stuff, but the ability of bacterial cells to actually work together in concert as a community is an important concept that has only been realized in the past 20 or 30 years, and that is bacteria do have the ability to send out chemical signals. They can actually work as a community rather than individuals. And so chemotaxis is a standard kind of mechanism by which you can begin to recruit cells and sort of organize them. Let's see. Was that it? What was it? I thought we had something about biofilms. We mentioned biofilms, and one of the things that chemotaxis can do is begin recruiting bacteria to form these larger structures. And these are actually an important way to think about the way some bacteria can self-organize themselves. All right, so lastly, and we probably should finish this on Monday, but I didn't. We have the different kinds of cell membrane and cell walls, and this is just a little minor point here, but we've got the eubacteria versus the archaebacteria, and if you recall, the archaebacteria are non-pathogenic, and so this is just sort of a minor point. You have slight different linkages between the fatty acids and the polar head group in the phospholipids. That makes a little bit of a difference between the way the bacterial cell wall or cell membrane is organized. Additionally, there is a little bit of difference in the archaebacteria's peptidoglycan and cell wall layer, a minor point. So this is one of the things that led to this distinction of not all prokaryotes are easily lumped into one great big thing that we call bacteria, but we can actually break that group into bacteria versus the archaebacteria. And it's a more evolutionary point, but who knows, you might find yourself growing interested in cell biology. Okay, so we're going to kick into actually some really fun now microbiology stuff that is more pertinent to the lab, and that is the way bacteria actually grow. So you may have heard, in human biology, this idea of what's called the cell cycle, the way cells divide. And in humans, in your standard person, we got cell division occurring at different rates. Some cells divide pretty frequently, like the cells in the lining of your mouth, intestinal epithelia. They're dividing probably once every 24 hours. And then you have other cells that are only dividing very rarely, very occasionally. Some cells really don't even go into the cell cycle much, like your neurons and things. And we also have cells in the body that are producing the gametes, the eggs and sperm, and they go through a very specialized cell division that is, of course, known as meiosis, as opposed to mitosis. In the case of bacteria, they've got a fairly straightforward cell cycle that is pretty much just the cells grow, they replicate DNA, the DNA is pushed to either side, and then you have the DNA replication, and then you have the division of the cell. The cell will pinch off, and you form two cells. So this is known as binary fission. One guy turns into two. And it is essentially what our cells are doing, our non-mitotic cells. But because it's a circular chromosome, single chromosome, it's a lot simpler. And it is way faster. So an average animal cell is dividing once every 24 hours. Your average bacteria cell, if you put it in the right conditions, is dividing once every 15 minutes. So again, don't race a bacteria cell in a cell cycle race, because they will beat you every time. I mean, there just is no way that we have a cell, and really any animal, is going to be able to beat this. We got so much more DNA, and the complexities of chromosomes, nah. Bacteria, they are cranking. And so they can divide really, really fast. Now, this kind of binary fission isn't the only way. We have a couple other microorganisms that operate in a slightly different way, and it's actually a useful way to kind of compare them. And that yeast undergo what is known as budding, where you have the next cell has the nucleus. It has the genetic material, but it's not the same size. So you don't have mom splitting into two in binary fission, or like you do in binary fission. Now you've got a little bud coming off. It's got the DNA in it, and it starts to grow, but it very distinctly is the little guy, and then eventually it'll pop off, so you have these little bud scars. There are also fungi that actually grow and spread through fragmentation, where pieces of almost like a stalk will break off, and then they'll begin doing their own kind of division. So binary fission, though, for bacteria is pretty much the standard. But you're just figuring we throw in, the exception that what might make the rule, whatever. Okay, so now when we start thinking about cell division, there are a couple of cool things to think about. In that, no matter bacteria, animal, plant, when you go through when you're going to divide, you are taking on a pretty serious responsibility to your children here. In that, you're going to replicate your DNA. And one does not replicate one's DNA in a trivial fashion, because errors in replication mean that the offspring are going to have troubles. Errors in replication might even screw you up. And so, consequently, and what we're doing here is a bit of what's called anthropomorphization. To anthropomorphize. Shit, I can't even. To place human tendencies onto non-human things. I do it all the time. Bacteria don't have these kinds of emotional notions, but they have evolved these control systems such that they do not go into cell division unless they have all the bits and pieces that they need. So, if we think of this, if you're going to divide, you have to accumulate a fair amount of stuff. You have to have sufficient individual nucleotides that when you start replicating your DNA, you don't run out of G, because that would be really bad. So you have to accumulate enough stuff that when you go into replication, you can just shoot right through it. It requires a fair amount of ATP to carry these kinds of manipulations out. The enzymes require energy. So you don't want to do this if you don't have sufficient energy. So what we do is, as a complex animal, we've got a lot of things going on, and in order to divide a cell, the cell has to recognize that there's sufficient nutrients, that there's sufficient reagents to do this, and then it'll go ahead and move forward. And it's a much longer process. It takes a fair amount of time to actually replicate all our DNA. Bacteria are going really fast, but they do have to make sure that they've got all the stuff needed. And so we have the cell cycle, and when we look at a way of breaking it down, and this actually is some, sort of a Nobel Prize winning approach to looking at the cell cycle, because it used to be just cells are dividing, and then when you start thinking about it, what we got is these phases of the cell cycle. And so we can think of the G phase as a gap. And this is where cells are growing and they are generating all the material that they need for carrying out cell division. And so this is pretty much a standard eukaryotic type guy. So you have the G1, which is the first gap. And then we take a pretty long time to make DNA. So this is very clearly a eukaryote because it's hours of time. And it takes you quite a while to replicate or to synthesize all the DNA. That's good. I mean, I want to take my time doing that because a mistake at this point could end up putting in the wrong nucleotide, which could then generate a protein that has the wrong amino acids, non-functional. This is a great way to get cancer. Take your time, do it right, make sure you have everything done, set just so. Then we have another gap here in which we can kind of rearrange things. And then finally we get to actual mitosis. And mitosis, you don't jack around with that, you do it pretty fast. So it's a little bit like launching the spaceship or a space shuttle, where you have internal holes. The countdown clock just stops because, all right, we want to make sure we have all these checks, that everybody has a go, no go. Can we proceed to the next stage? And if you can, all right, we go. Now, there are some eukaryotic cells that exit the cell cycle. They're not playing the game anymore. It's what's called G0. Neurons do this. So your neurons are not keen on replicating or dividing. There is various sorts of controversy in therapy, but neurons in general don't do a great job of replicating very fast. But a standard epithelial cell, there you go. Now, compare this to a bacterium, and a bacterium is going really fast. They are just zooming through that cell cycle. They don't take the gaps the same way. They are going to be moving through the cell cycle at a much faster clip. And in part because bacteria, they're probably not quite as careful, but they also have a much faster ability to go through the cell cycle, and they're kind of living in the moment. If they got enough material, they go. When things get sloppy, there's not enough material, enough food, they'll just stop. All right, so another aspect of bacterial cell division that we compare to eukaryotic division is that bacterial cell division is pretty much asexual. That is, you're building clones. So this binary fission, 1 becomes 2. And you start out with a single bacterium, which actually when we start playing around in the lab this afternoon, just imagine this, you're going to be diluting bacteria out to where you got a single bacterium floating around in a solution. When we plate out bacteria, or streak it out, actually when we plate it out, you'll be pouring a solution under the plate, spreading that solution out, and then tomorrow we'll come in and we'll see individual colonies. Those colonies started from one single guy. And so this is a characteristic of these kinds of single-cell organisms, is that one guy, one cell, can begin producing a colony. Now, in sexual reproduction, we've got, rather than 1 becoming 2, we have a female, an egg, and a male, a sperm, fusing and producing a unique organism. And this allows for a lot more genetic variety. So clones, like bacteria, don't have much in the way of genetic variation. But sexual reproduction, you always are having this new sort of introduction and scrambling of new kinds of gene types and things. So you can do all kinds of things with sexual reproduction that you can't do with clonal or asexual reproduction. But, asexual reproduction is a good bit simpler anyway. So now, when we look at the bacterial growth, you can measure it in a number of ways. You can actually count the number of bacteria, which is rather tedious. A simpler way to do it is actually to look at what's called the optical density, i.e., how cloudy is it. And when we are looking at the number of bacteria, over time, we can develop this kind of curve, and that we have the initial phase, and if you want to think of this as the initial phase of the infection, the lag phase, it's where you got the initial infection, or the initial inoculation, and the bacteria are sort of figuring out what the conditions are. They begin to start assessing what kinds of food there is, what the different sugars, what the different nutrients are, what do they need to do to start breaking these things down, and they begin to set up shop. And you then get into the log or exponential phase. This is where we have growth at a logarithmic or exponential kind of rate. So you're going to, n is the number of generations. It can't continue because you're just going to end up running out of stuff before too long. But it goes pretty fast. Medically, this is probably before you know you got anything, and you're beginning to establish a pretty big infection now. So then, you get to a spot that we call the stationary phase. And this is going to be dictated by a lack of nutrients very often. But additionally, you have all these guys floating around in the media or in the wound, and they are dumping out sort of the equivalent of bacterial pee, various metabolic waste products, and it can actually get to a spot where the waste products become somewhat lethal. And so once you get to this spot, the cells begin to adjust, and essentially what we have is the number of new bacteria is equivalent to the number of bacteria that are exiting this life that are dying off. That maintains the same number. So this is a stationary phase. Now, in this case, we have the production of an abscess, a biofilm. You're probably starting to see symptoms now at this top spot in the curve. All right, so then we have the death decline phase. You've eaten up all the food. You have peed so much into the media that no one can survive. And after a time then, the loss of bacteria is actually greater than the new bacteria coming up. You end up medically feeling better, symptoms are improving, but you haven't knocked the infection back all the way, but you're probably, hopefully you're on the road to getting better if you can survive that. So this is a standard kind of growth curve. When we're working with bacteria, what we typically will do is start a liquid culture, let it begin growing and shaking to put lots of oxygen into it. These are aerobic bacteria. And you'll have this big flush, and then it'll hang there. And typically what you do is you start a culture, this afternoon at 3, let it grow overnight, and hence it is known as an overnight culture. And the next morning you come in, you have a saturated culture. You have hit the stationary phase. There are some experiments in molecular biology where we want to have things in this part of the exponential growth phase. You want the bacteria to be divided as fast as they can, particularly if you're going to be manipulating them by putting DNA in, or if you want to lyse them, you want to break them apart. If they're dividing really fast, their cell walls aren't real stiff and real thick, and so you want to catch them in this exponential growth phase. But generally, that's like within 2 or 3 hours of starting the culture. I mean, it's pretty early on. And once they've hit stationary, they start to get really a lot harder to break apart and manipulate. So that is a standard kind of growth track. Now, this is a list of definitions that I encourage you to write down and commit to memory. It's a way of thinking about how the different bacteria gain their nutrition. And all right, so we as animals are what are known as heterotrophs. One of the things we start comparing animal life with bacterial life is that pretty much if you're an animal, and particularly if you're a vertebrate, well, you're a brother, you're a sister. I mean, the difference between vertebrates is insignificant compared to the similarities. That is, if you got a spine, I mean, you pretty much are a member of my tribe. And that even holds for fish, man. If you got a spine, welcome to the club of vertebrates, and the differences between us is so small, maybe I shouldn't be eating you, and you shouldn't be eating me. So a dog is a person, a cat is a dog, horses, we're all the same thing. If you're a vertebrate, I'm down with you. That said, we don't recognize just how bizarre and how varied life can be. Now, when we start thinking about what it takes to actually operate, you got to have food. Yeah? Okay. The food that we eat, happily, is carbon-based. If you find yourself eating a lot of stuff that's not carbon-based, you need to really seriously evaluate your diet. Like what? You're eating dirt, you're eating glass, you're eating nails, metal shavings. Don't do that. We eat carbon. We eat bread and vegetables. We eat other living things, bacteria and yogurt. So, we're getting our source of energy and our source of carbon and our source of electrons all from the same thing. All from the same material that you're eating. Not all living systems do that. But that's one of the things that's kind of hard to get your head around is that, I mean, yeah, I know plants are getting energy from sunlight, but there's some really weird stuff out there. There are weird organisms out there. So if we break down why you eat, you have to have a source of energy. And in phototrophs, that is sunlight. And so this is guys like plants. They carry out photosynthesis. I don't know if you had your basic biology talking about photosynthesis, but the energy from sunlight is what's driving the chemical reactions and driving the energy production. For other kinds of organisms, like myself, we're getting energy from chemical bonds. We're breaking bonds and extracting energy from that. All right, so we can't photosynthesize. Another thing is that we have to have a source of electrons. I think we went through this in terms of our metabolism, anabolic versus catabolic. And if we haven't, we're going to get to it later. That you got to have, if you're going to be building up these polymers, you have to have a source of electrons to make these new covalent bonds. So all organisms need fresh electrons coming in all the time. And the source of those electrons can be a variety of things. So lithotrophs are using non-carbon-based molecules to extract their electrons. So you're taking iron and pulling electrons out of it. You're taking hydrogen sulfide and pulling electrons from it. Ammonia, a variety of things. Now, organotrophs are using organic molecules as our source of electrons. And so what we got here is a convenient one-stop shop, is that when you shove that donut in your mouth, you're getting your source of energy, your source of electrons, and your source of carbon all in one go. There are a lot of organisms out there, bacteria anyway, that they got to have a grand buffet. They have stuff they get their energy from, they got stuff they get their electrons from, and then they get their stuff they get their carbon from. You know, so it's like the ultimate in a picky eater. So organotrophs, we're getting organic chemicals for our source of electrons. Lastly, the source of carbon. And again, for us, you eat carbon. So we were one-stop shoppers. But there are organisms that are able to bring in carbon into their body by taking in inorganic carbon in the form primarily of carbon dioxide. So plants do this. You don't see plants eating much. Plants will chew on bugs if you're a carnivorous plant, but not very many are. But they're probably getting the nitrogen that way. Plants get their carbon by pulling in carbon dioxide. Bacteria do the same thing. And then we again have that heterotrophs, they're pulling in the carbon from the organic chemicals. So consequently, we have these names that we can put together that are a portamento or a combination of the energy source, the electron source, and the carbon source. And so we've got a photo-organo-heterotroph, which I guess there is things that are that way. This would be, well, like some kind of photosynthetic plant that is eating people. Don't get around those. We have then the chemical compounds as the energy source, and they can be organic, where we're getting our electrons, and then we can have organic carbon source. This would be us. We are chemo-organo-heterotrophs. Then you got our various plant friends that are chemo-organo-autotrophs. Actually, their source of electrons is not organic. What are these guys? All right, so this is what a plant would be. A chemolitho-autotroph. Yeah. All right. I'm probably not going to throw that much like this at you, but for sure, the definitions of these guys is important. And being able to, once you can find them well, you start linking them together. So it only is an issue because not everything eats the way we do, but I'm glad to be a chemo-organo-heterotroph in that I don't have to eat a variety of different things. So bacteria that are in our body and most pathogenic bacteria are optimized to grow at body temperature, 37 degrees. But there are not, that's not the only bacteria. And so if we're going to be talking about strictly pathogenic bacteria, then this slide is probably not all that useful. But hey, we're going to do a little bit more stuff. And there are bacteria that can grow in extremely cold temperatures. And we got a couple more definitions here, in that we have the trophs versus the phials. So a psychrophile is cold requiring, whereas a psychotroph is a guy that can grow in the cold but would prefer not to. The moderate range, mesophiles, they are probably limited to this temperature range. They're not going to grow outside that range, and they're usually going to be dead. So there are bacteria that reside within us. They probably are not going to be very happy outside. Below freezing, for sure not. And certainly, once you get above, a strong fever, 110 degrees, they're probably dead too. So mesophiles typically are the kinds of pathogenic bacteria, and they do have a fairly narrow range. Unfortunately, our cells have a fairly narrow range as well. So then we have heat-tolerant bacteria, thermotolerant versus heat-requiring thermophiles. And these are the term we use for guys that can grow at temperatures greater than 40 degrees centigrade. So your body temperature is running at 37. There's not that many disease-causing pathogens that are going to be all that heat-tolerant. Not off the top of my head anyway. And then there are some, though, that are hyperthermophiles that are growing in essentially hot springs, growing at temperatures greater than 70 degrees. And so, I don't know if you ever live in the centigrade Fahrenheit world, or kind of combine the two, but I always try to have these landmarks of different temperatures. And so, hopefully you know that 0 degrees centigrade is freezing, 32 degrees. Right now, it's like, minus 10 or so centigrade outside. Body temperature, that is 98.6 Fahrenheit, that's 37 degrees centigrade. And your hot water tank at your house, if you like a really hot shower, is probably running about 130, 140. Water much above 110, this is Fahrenheit, is not all that comfortable. You can probably sit, I don't know if you've ever been like either in a really hot tub or a hot springs or in a spa where they've got these hot, really hot spots. 110 degrees, man, it's that kind of feeling that if you move, it starts to sting. And you can probably get into 110 degree water, but it is really, really hot. And much above 110 degrees, you're going to touch it and you go, damn. So your hands are pretty tough. I don't know, you have hot water heaters that are cranked up, depending on where you live. I mean, our hot water heater at the house runs about 140, which means that at full blast hot, you can't put your hands under the hot water. So these are temperatures that we're talking about. And hyperthermophiles at 70 degrees, damn, that's like running at 180 degrees. That's really, really hot. And these bacteria, they're able to operate that way. So the main problem is that protein structure is based on those weak-ass bonds, and temperatures much above, at least for us, body temperature, make it so the protein loses structure, they denature. But these guys have proteins that are evolved to actually operate with these very high temperatures. And their membranes are pretty squirrely too, because they don't kind of just dribble off into nothing. But again, not pathogenic. Now, when we start talking about further environmental conditions, the issue of oxygen becomes a really cool one too. And this actually does have some stuff that we can relate to disease. In that oxygen is a critical molecule for our life. Try living for more than 2 or 3 minutes without oxygen. I dare you. Yo.
Well, like back to Sly, what's like the structurally different, like that's so different that they can handle like the 180-degree temperature?
Well, they're going to have virtually all saturated fatty acids. Their wall structure is really tough, that they're able to handle the temperature, and so the membrane's quite gooey and wobbly, but they're able to sort of maintain their structure that way. And the proteins end up having hydrogen bonding structures that are tucked in and held together really, really tightly. Because certainly there are hydrogen bonds that are going to take boiling or near boiling to break them apart. I've never even worked with the thermophilic bacteria. I work with enzymes from thermophilic bacteria, and those are hilarious in that they can be boiled, and then you bring the temperature down just a bit, and they start working at optimum, at their maximum rates. And they're not very functional at room temperature. So these make for some pretty cool, the technology behind the test for the coronavirus is the PCR, the polymerase chain reaction, which depends on having a sample that you boil like 34, 35 times. And so you have to have this thermotolerant enzyme in order to have that work. So in terms of psychrophilic guys, there are some systems and some enzymes that I've used in the molecular biology lab that are psychrophilic. Really, primarily what that means is you can denature them pretty easy. Because there are, even within our bodies, there are proteins that are pretty damn tough. They won't work at boiling, but as soon as you cool the temperature down, bring it back down to 37 degrees, they'll fold back up and they'll get back to work. Most of the proteins in our bodies, when you heat them up, they are done. But there are some that'll keep on working. And so psychrophiles end up having proteins that when you heat them up, they pretty much dead. All right, so again, we can't operate without oxygen. And so, again, there are no animals that operate without oxygen. And so when you think about the animal kingdom, this just doesn't make sense. But when we start talking about bacteria, man, there's some really weird bacteria. There are guys that are obligate aerobes, and just as a little note, we are obligate aerobes. They have to have O2 for this idea of cellular respiration, which hopefully you had a little bit of it like in Essentials. This is what we need to have to make ATP. Microaerophiles are kind of weird guys. They require oxygen, but they have to have it in small amounts. All right. Then we've got facultative anaerobes. And a lot of bacteria can work like this, in which they really need oxygen to actually divide fast and really get going, but they can operate without it. And they go into either anaerobic respiration or fermentation. Again, we are not particularly facultative anaerobes because you can't live without it. But when you're exercising really hard, you begin to make lactic acid in your muscles because your muscles go into oxygen debt. And if you did this full body, you go into lactic acidosis and you're dead. What you can do is carry out your exercise while breathing pure oxygen, which is kind of cheating, I guess. But that's like in the NFL, you can have oxygen on the sidelines. And so you might, as a great big linebacker, grabs an interception and runs down the field. He gets back to the sidelines and he puffs on oxygen for a while. I think in D3 you can't have oxygen on the sidelines even. What you're doing is you're repaying your oxygen debt really fast. Same thing you do is you breathe hard. Okay, it's more fun to breathe pure oxygen, but whatever. But there are a lot of bacteria, and actually yeast can do this too, that they'll grow way faster in the presence of oxygen, but they'll grow without oxygen. And then we've got some odd guys that are obligate anaerobes. They find oxygen to be poisonous. Now, interestingly, this is a cool thing to think about. There are pathogenic bacteria that can be obligate anaerobes. And these are guys that are only going to thrive in a wound that is really deep. And so the bacterium that causes tetanus is an obligate anaerobe. For that matter, so is anthrax. And they grow in conditions in which there is no oxygen. Now, they will hang about in an oxygen atmosphere, but they're not going to start dividing. And so if you get a deep puncture wound, particularly when it seals back up, that little area actually can start to go anaerobic. And when it does, it allows those bacteria to start growing up again. So oxygen does generate some fairly corrosive molecules. So we use oxygen, but we also have to deal with the after effects of using it, and that there are oxygen radicals that are damaging to our bodies. So you get the oxygen radical, you have different kinds of enzymes that can relieve this. This is things like hydrogen peroxide. You can break that apart into water and O2 if you got two of them. There are a variety of different kinds of enzymes that will take oxygen radicals and generate some kind of non-damaging molecule. When you start getting into metabolism, you'll see that when we use oxygen as the final electron acceptor for aerobic respiration, you take O2 and we're going to make water, H2O, which means that every time you do this, you're splitting the O2 molecule. And for a moment, you're left with an O1, or the chemists would have us call it an O1 half O2, regardless. It is this oxygen radical. This is the active ingredient in hydrogen peroxide. It is a phenomenally strong bleaching and corrosive agent. And our cells are being exposed to it constantly. And the only way you can reduce that exposure would be to reduce the amount of food you're eating and reduce the amount of calories you're eating. And it becomes an interesting game because obviously if you reduce your calories too low, then you're dead. But calorie restriction might actually work as a life extension diet because you are reducing the amount of oxygen radical that you're being exposed to. All right, so we got then these different kinds of environmental conditions, and I think we're going to stop here. Yeah, we'll stop here on getting the pH and some weird guys that can survive in acids. And I'll see you at 1. So go grab lunch. And 318 upstairs, third floor. I'll be hanging around. I'll see you then. All you need is your lab protocol and pencil and paper and stuff. We pretty much, just because of safety's sake, have you put all your gear on the side. So don't bring in anything too much if you don't have to. And I'll catch up with you then.